Hi, welcome to the part 2 of cellular respiration. So in this part 2 of cellular respiration, we'll discuss about the different stages that happen in cellular respiration for aerobic respiration. Now, before we look further into the different stages in cellular respiration, it's important for you to remember back about the structure of mitochondria. This is because most stages that happen in cellular respiration actually takes part inside the mitochondria. So first here, the outer layer of the mitochondria is known as outer membrane. Within the outer membrane and the inner membrane of the mitochondria, there is a space which we call as intermembrane space. And this folded structure here is what we call as cristae, and cristae here is actually surrounded by inner membrane, and this part inside here, inside the cristae, is what we call as the mitochondrial matrix. The main objective of cellular respiration is to generate ATP by breaking down glucose. Phosphorylation is a process of forming ATP by addition of phosphate to ADP. There are two types of phosphorylation in cellular respiration. First one is oxidative phosphorylation and the second one is substrate level phosphorylation. Oxidative phosphorylation involves the formation of ATP by adding P to ADP, but this process requires a driving force. This driving force is the result of teamwork between electron transport chain and chemiosmosis. ATP is produced by oxidation of NADH and FADH2 and subsequent transfer of electrons along electrons transport chain. As a result, there are H+ or proton being pumped across the mitochondrial inner membrane to the intermembrane space, and this will generate an electrochemical gradient. This electrochemical gradient is what will be used to power ATP synthase to produce ATP by adding phosphate to ADP. On the other hand, in substrate level phosphorylation, the process does not require any driving force and it only requires a direct transfer or donation of inorganic phosphate to ADP to form ATP, or transfer of phosphate to GDP to form GDP. There are three major stages that happen in cellular respiration with the presence of oxygen, which are glycolysis, Krebs cycle, and oxidative phosphorylation. In glycolysis, glucose is broken down into two molecules of pyruvate. Hence, glycolysis can also be known as splitting of sugar. Glycolysis happens in cytoplasm and has two stages, which are energy investment phase and energy payoff phase. Based on the net reaction in glycolysis, one molecule of glucose will be converted into two molecules of pyruvates along with the production of 2 NADH, 2 ATP and 2 molecules of water. The NADH will need to enter mitochondria to go to electron transport chain to transfer electron. This will then generate ATP. The only stage that will happen with or without oxygen is glycolysis. In the first stage of glycolysis, which is energy investment phase, ATP is used to phosphorylate the glucose, or we can also say that to activate the glucose. This is to prevent the glucose from leaving the cell. So the first one is phosphorylation. ATP will come and donate its phosphate into glucose, turning the glucose into glucose 6-phosphate. Then this glucose 6-phosphate will undergo isomerization into fructose 6-phosphate. Basically, it is only the rearrangement of the structure. And then the third one is phosphorylation. So another ATP will come and donates its phosphate group into the fructose 6-phosphate, and now they will become fructose 1,6-biphosphate. It is now a structure with 6 carbon but with 2 phosphate group. And this fructose 1,6-biphosphate with 6 carbon will need to be cleaved. Okay, and this will produce 2 molecules with 3 carbon. The first one is dihydroxyacetone phosphate or DHAP, and then the second one is glyceraldehyde 3-phosphate or G3P. But this DHAP will need to undergo isomerization to produce G3P. At this stage, from 1 molecule of glucose just now, there will be 2 molecules of G3P at this point. The 2 molecules of G3P just now will next be undergoing oxidation. In oxidation, NAD+ will come and accept electrons and also H+ from the G3P, and they will be reduced into NADH. At the same time, there is also an inorganic phosphate that will come and bind to the structure. So the G3P just now will form 1,3-biphosphoglycerate. 1,3-biphosphoglycerate will next be undergoing substrate level phosphorylation. In this step, ATP will be produced. The phosphate from the 1,3-biphosphoglycerate will be donated or transferred to ADP to produce ATP, and this will produce 3-phosphoglycerate. 3-phosphoglycerate will undergo isomerization, rearrangement of structure to form 2-phosphoglycerate. 2-phosphoglycerate then will remove water molecules from their structure, and this is called as dehydration. This will form PEP or phosphoenol pyruvate. PEP plus will undergo another substrate level phosphorylation. Another ADP will come and receive phosphate from the PEP to form ATP. At the end of the stage of glycolysis, there will be 2 molecules of pyruvate from 1 molecule of glucose, and this is the energy payoff phase where ATP will be produced. Glycolysis generally have 2 phases: energy investment phase and energy payoff phase. In energy investment phase, 2 ATP are used, and in energy payoff phase 4 ATP are formed. Other than the 4 ATP, there are also 2 NADH being produced, and from 1 molecule of glucose, by the end of glycolysis, you will get 2 molecules of pyruvate. The net production of ATP here from glycolysis is actually only 2 ATP, but not 4 ATP because the other 2 ATP will be used to replace the 2 ATP that were used in the energy investment phase. So you will only have 2 ATP along with 2 NADH and 2 pyruvate by the end of glycolysis. Okay, so tadi glycolysis akan menghasilkan NADH and juga 2 pyruvate, and also there is 2 ATP. So the NADH just now akan masuk ke dalam electron transport chain untuk menghasilkan ATP. melalui oxidative phosphorylation. So here, pyruvate tadi daripada glycolysis. Okay, from the glycolysis, this pyruvate, here the pyruvate, okay, one molecule of pyruvate will enter the mitochondria. But as this pyruvate enter the mitochondria, it will undergo several changes. Okay, so this pyruvate will crosses the mitochondrial membrane through transport proteins. First, the pyruvate, okay, they will undergo decarbosylation, di mana carboxyl group akan dikeluarkan, and they are being removed in the form of carbon dioxide. And then this leave the pyruvate with 3 carbon just now, becoming only 2 carbon. So the 2 carbon is oxidized, okay, meaning it loses or it transfer dia punya electron to NAD+, and this will form acetate. And acetate will combine with coenzyme A to form acetyl-CoA. So acetyl-CoA will have 2 carbon, and this from one molecule of glucose, okay, that from at the end of glycolysis will produce 2 pyruvate, and through link reaction here, okay, they will form 2 acetyl-CoA, and this acetyl-CoA will enter Krebs cycle or citric acid cycle. So this link reaction involve oxidative decarbosylation. There is oxidation and removal of carbon, okay, or carboxyl group. And the enzyme that involved in this reaction is pyruvate decarbosylase. Acetyl-CoA from the link reaction just now will next enter Krebs cycle. Krebs cycle normally happen in mitochondrial matrix. Krebs cycle, or also called as citric acid cycle, will complete the breakdown of pyruvate to carbon dioxide. Based on the net reaction of Krebs cycle, one molecule of acetyl-CoA will produce the production of 3 NADH, 1 FADH2, 1 ATP, and also 2 carbon dioxide. This NADH and FADH2 will then need to enter electron transport chain. In electron transport chain, both NADH and FADH2 will undergo oxidation to generate ATP production. At this point, you must make sure you understand that from one molecule of glucose, how many turns does the Krebs cycle will happen, and what are the main products. So this diagram shows one whole cycle of Krebs cycle. So acetyl-CoA will enter Krebs cycle, and there are few productions of NADH, ATP, FADH2, and also NADH. We will go through this Krebs cycle step by step. So here, acetyl-CoA with 2 carbon will enter Krebs cycle, and this acetyl-CoA will bind with oxaloacetate with 4 carbon. This step is known as condensation. 2 carbon acetyl-CoA will bind with 4 carbon oxaloacetate and form 6 carbon citrate. Oxaloacetate will bind to the acetyl group. The coenzyme A will be displaced, and this free coenzyme A will go back into the link reaction. Next one is the 6 carbon citrate will then undergo isomerization. A molecule of water is also added and then removed, and this will form 6 carbon isocitrate. The 6 carbon isocitrate will next undergo oxidative decarbosylation. Carboxyl group are removed from the isocitrate in the form of carbon dioxide. and at the same time NAD+ will come and receive electron and H+ from the isocitrate. The NAD+ will be reduced into NADH, and this will form 5-carbon ketoglutarate, alpha ketoglutarate. This alpha ketoglutarate will also undergo another oxidative decarbosylation, and carbon dioxide is removed, and it is also oxidized by NAD+ to form NADH. Then coenzyme A will come and bind on the structure, forming a 4-carbon succinyl-CoA. Next, the succinyl-CoA will undergo substrate level phosphorylation. The 4-carbon succinyl-CoA is displaced by a phosphate group into a 4-carbon succinate. The phosphate group is then transferred from the structure to a GDP, forming GTP. This GTP will then donate its phosphate group to ADP to form ATP, and the process of forming ATP here is what we call as substrate level phosphorylation. Next, there will be oxidation. The 4-carbon succinate will undergo oxidation, in which electrons from the succinate and also H+ will be transferred to FAD+, and this will reduce FAD+ into FADH2, and this will produce a 4-carbon fumarate. The 4-carbon of fumarate will next undergo hydration. In this hydration process, 1 molecule of water will enter the structure, and this will form 4-carbon of malate. This 4-carbon of malate will next undergo oxidation, in which electron and also H+ from the malate will be transferred to the NAD+. NAD+ will then be reduced into NADH, and this will form the 4-carbon oxaloacetate just now that previously have bind into the acetyl-CoA. Basically, the last stage just now is actually to form the 4-carbon oxaloacetate. This 4-carbon oxaloacetate will normally bind to the 2-carbon of acetyl-CoA from the link reaction to form back the 6-carbon, and the cycle will repeat itself. And from this cycle, you can see that from 1 molecule of pyruvate, which will form 1 acetyl-CoA, and here after this acetyl-CoA enter the citric acid cycle or Krebs cycle, there will be 2 carbon dioxide produced, 3 NADH, 1 ATP, and also 1 FADH2. So here what we can get is that from 1 acetyl-CoA, after it has entered the Krebs cycle, there will be 1 GTP or ATP directly being produced by substrate level phosphorylation, 3 NADH, and 1 FADH2. But how about if there is from 1 molecule of glucose? How many GTP or ATP, how many NADH, and how many FADH2 will be produced? Can you guess? So you must remember correctly that from 1 glycolysis, from 1 molecule of glucose, there will be 2 molecules of pyruvate being produced, and this pyruvate will enter link reaction and produce acetyl-CoA. So there will be 2 acetyl-CoA by the end of link reaction, and these 2 acetyl-CoA will then enter Krebs cycle. So meaning here is that if it is from 1 molecule of glucose, there will be 2 GTP which will next form directly ATP by substrate level phosphorylation, 6 NADH and also 2 FADH2. Now, all the NADH and FADH2 which were produced from glycolysis and also Krebs cycle will need to enter oxidative phosphorylation, in this oxidative phosphorylation, the NADH and FADH2 will need to transfer their electrons to electron transport chain, and this electron will power the ATP synthesis. Oxidative phosphorylation has two parts, which are ETC or electron transport chain and chemiosmosis. Electrons will drop in free energy as they go down the chain and are finally passed to oxygen, forming water. Meaning here is that the final electron acceptor after electron transport chain is actually the oxygen. Krebs cycle and also oxidative phosphorylation can only happen in the presence of oxygen. Oxidative phosphorylation is a series of teamwork between electron transport chain and chemiosmosis. The first part is electron transport chain, or we can just call as ETC. ETC are located in mitochondria. To be exact, the electron transport chain can be found on the inner membrane of mitochondria. As NADH and FADH2 arrive from glycolysis and also Krebs cycle, their high energy electrons will be transferred to the electron transport chain, and they will move from one acceptor to another acceptor, which means there is a series of reduction and oxidation that happen in electron transport chain. ATP from oxidative phosphorylation here is produced by using energy required as the electron is passed along from one acceptor to another acceptor. This is the reason why the ATP produced here is called as oxidative phosphorylation. Electron transport chain is directly attached on the inner membrane of mitochondria. In electron transport chain, there are few members including flavoprotein or FMN, coenzyme Q, several iron sulfur, and also a group of proteins that contain iron, or we call as cytochrome. For example, cytochrome B, cytochrome C, and even the last one here is cytochrome A3. There are also 4 large protein complexes as acceptor in this electron transport chain, which are protein complex 1, protein complex 2, protein complex 3, and protein complex 4. And at the end of this electron transport chain, there are oxygen, and oxygen here has the function as final electron acceptor. In electron transport chain, the NADH will enter the electron transport chain through protein complex 1. NADH will donate its electron to protein complex 1, and this electron will move from one electron acceptor to another electron acceptor. And meaning here is that if one electron acceptor accepts the electron, they will become reduced. And if the electron acceptor donates or transfers the electron to the next electron acceptor, they are oxidized. And here we can see that electron from NADH will first enter complex 1, and then they will pass to Q, and then into cytochrome complex 3, and then cytochrome C, finally to complex 4 before these electrons are then being accepted by oxygen. For FADH2, the electron from FADH2 will not enter from complex 1, instead they will enter from protein complex 2, and then this electron will next be passed to Q, and then into complex 3, and then cytochrome C complex 4, and also finally they will reach the oxygen. And if the oxygen will accept the electron from the electron transport chain, they will combine with H plus and then will form water. At the end of this electron transport chain, the electrons will finally reach oxygen, and if the electrons reach the oxygen, there will be a chemical reaction between hydrogen and oxygen, and this will produce water. And the reason why the electron is able to move from one electron acceptor to another electron acceptor along the electron transport chain is because electron acceptor has high electron affinity than the electron donor. But wait, at this stage the electron transport chain does not generate ATP directly. Hence, there is still something missing here. ATP generation in oxidative phosphorylation can only happen with the help of chemiosmosis. This electron transport chain is actually located or attached on the inner membrane of mitochondria. And if the electrons are donated, for example here from NADH into complex 1, this electron will move from complex 1 to Q, complex 3, and then to cytochrome C, complex 4, and then finally it will reach oxygen. Whereas FADH2, the electron will only enter from protein complex 2 and into the Q, and so on until it reaches the oxygen. Here, every time when the electrons are passed from one electron acceptor, or as they pass from one protein complex to another protein complex, H plus will be pumped. This H plus from the mitochondrial matrix will be pumped through the protein complex into the intermembrane space. Okay, so this is the intermembrane space. From this protein complex, only protein complex 1, 3, and 4 are able to pump H plus into the intermembrane space, but not the protein complex 2. Okay, so as a result, there will be high H plus that will build up at the intermembrane space. This is what we call as the generation of electrochemical gradient. It is dangerous for it to have high H plus at the intermembrane space because this will reduce the pH. Okay, so you remember high H plus concentration will reduce pH. Okay, so accumulation of protons in the intermembrane space will create a proton gradient. So how they should fix this is by the H plus in the intermembrane space need to move back inside the membrane and back into the mitochondrial matrix. This is with the help of ATP synthase. Okay, so this is ATP synthase, and ATP synthase here is a transmembrane protein. H plus will then move back into the mitochondrial matrix by passing through ATP synthase, and this is what will generate the formation of ATP by adding inorganic phosphate to ADP. So the flow of protons through ATP synthase will provide energy for ATP synthesis. This is by addition of phosphate into ADP. From the whole process, you can find out that the oxidation of one molecule of NADH will give 3 molecules of ATP, whereas one molecule of FADH2 will only produce 2 molecules of ATPs. This is due to how the electrons enter the electron transport chain. NADH will only enter from complex 1, whereas FADH2 will only enter from complex 2. Based on the overall reaction, from 1 molecules of glucose, the glycolysis can actually already produce 2 ATP. This ATP produced from glycolysis is from substrate level phosphorylation. Other than the 2 ATP, there are also 2 NADH from glycolysis. This NADH will need to enter mitochondria so that they can donate their electrons to electron transport chain for generation of ATP. So from this generation of ATP, by using oxidative phosphorylation, 2 NADH will produce 6 ATP. It is only times 3. And the reason why you need to times 3 is, remember just now, oxidation of one molecule of NADH will give 3 ATP, whereas one molecule of FADH2 will give 2 ATP. Next, formation of acetyl-CoA or through the link reaction. In link reaction, there will be 2 NADH, and this NADH will also need to enter electron transport chain. Hence, you will get 6 ATP. Under Krebs cycle or citric acid cycle, there are 2 ATP that is directly produced from substrate level phosphorylation. The 6 NADH and 2 FADH2 will need to enter electron transport chain, and respectively they will produce 6 NADH will produce 18 ATP, whereas 2 FADH2 will produce 4 ATP. Hence, the total ATP from the overall reaction of aerobic respiration is 38. And this 38 will not be able to be the same in muscle cell and neuron cell, which only will produce 36 ATP. The difference is in the NADH from glycolysis. This NADH is produced in cytoplasm, but they will need to move into mitochondria so that they can reach the electron transport chain. In order for this NADH to enter the mitochondria, they will use different type of shuttle. So this different shuttle mechanism is what makes the difference whether it is 38 or 36 ATP. Do you know what is cyanide? Have you all heard about the danger of cyanide? Cyanide can be dangerous, especially if it is exposed to the environment. Cyanide is widely used as poison. But why is this cyanide can be harmful to human? This cyanide can inhibit respiration. This is because cyanide is actually an electron transport chain inhibitor. Cyanide can inhibit the last cytochrome in the electron transport chain, which is cytochrome A3. If cyanide binds on this cytochrome, then the electrons will not be able to reach the oxygen, and all the electron acceptor will be filled with electrons, as the flow of electrons will be blocked. Other than cyanide, there are also other examples of electron transport chain inhibitors. For example, carbon monoxide, antimycin A, which is a fish poison. So please, students, please read on the different inhibitors of electron transport chain and how do each of them can do the inhibition on electron transport chain.